All right, people, how are you? Today we are going to be looking at a subtopic called secretions of the GIT. All right, so we did look at the anatomy, we also did look at the overview, we also did look at the motility aspect of the GIT. But now we are going to focus on what we call secretions of the GIT in the mouth, the stomach, small intestines, and the large intestine. All right, so as you can see right now, is what we call the three major salivary glands that you can see on the screen. So you can see there that we have got what we call the parotid, there's what we call the submandibular, and there's what we call the sublingual gland, right? So these three culprits have got a specific function or a major function, which is to produce secretions that enable or facilitate the process of either physical or chemical digestion. What I mean by these two terminologies is that because you have to be very familiar with the nomenclature of the GIT or the nomenclature in physiology. So now when you talk about these two, you're going to find that when you talk about the physical aspect, that's where now saliva comes in terms of lubrication, making the food soft so it can easily be broken down and exposing the surface area, right? Then the chemical aspect comes in when these glands produce what we call enzymes and chemicals that can start the process of digestion. So now let's begin with what we call the parotid gland. So this parotid gland, as you know it, is the one that is responsible for the production of large quantities of these chemicals, enzymes, or secretions, right? Then we have got these two subordinates or these two other, in Bemba we can say even like a parcel, which are known as the submandibular or the sublingual. They can also facilitate in the process of starting digestion, either physical or chemical. But the secretions they produce are in a lesser quantity as compared to the parotides. Such questions usually come in best answer or what we call MCQs. All right. So you can see here that we are going to be talking about two things under secretions. There's going to be what we call the exocrine and the endocrine. But we are going to focus so much on what we call the exocrine. Why are we focusing so much on the exocrine? Okay. We are focusing on that because that's what consists of the GIT, while the endocrine comes in as a helper. Remember, I did talk about in our previous sessions or in the introduction, we did talk about what we call the enteric nervous system. So the enteric nervous system is also known or referred to as the gut brain or the brain of the gut. Okay, meaning the gut or the GIT has got a brain of its own. It's able to make its own decisions without your conscious or without you putting anything there. So those are the things that you're going to be discussing under this subtopic. We are also going to involve what we call the enteroendocrine. These are chemicals which are part of the endocrine system that are going to be produced to enhance or facilitate the process of digestion. So what you are seeing here is a pair of glands. Yeah, so as you can see, they've got, as you know, exocrine, they're supposed to have glands, all right? So whatever is secreted by the parotid gland goes into the mouth. Whatever is produced by the submandibular gland goes into the mouth. Whatever is produced by the sublingual goes into the mouth via their duct. Okay, all right. So now these are the three pairs: the parotid, the sublingual, and submandibular. All right. Functions of saliva. We're going to realize that under this, saliva has got functions like lubrication. Okay, so lubrication helps in the fact that in order for you to swallow food, it needs to be lubricated. I usually give an example when you get something that is very fine ground, or that is very fine. For example, you get powdered milk, you get maybe soya beans, ground soya. When you put it in your mouth as long as it's not properly lubricated. You cannot swallow it. You can, but there will be a lot of difficulties in swallowing because the due process of lubrication has not taken place. So that's one of the functions of saliva. The other thing is that it's able to cleanse the oral cavity. All right, meaning it's able to clean for example, if you take in things that you don't want, for example, even without taking water, if when you are young, you are given a very bitter drug like chloroquine, or maybe Fancida, or anything that is bitter, you put it in your mouth, all right? You're going to realize that once you ingest that chemical, that product, even without water, after some time, the taste in your mouth is going to normalize. That's what we call cleansing, all right? So also, once you're having a meal, the saliva, together with the help of the tongue, is able to clean your mouth. That's also referred to as cleansing. It can also go further by dissolving chemicals. For example, the only way you can feel taste is because of the saliva that is in your mouth. Because the food that you eat will dissolve in saliva. Then your taste buds are going to pick them up. When you go further in terms of studying physiology, you go and do neurophysiology and the spatial senses, you're going to realize that taste buds work together with saliva, right? They are able to pick up the receptors that are protruding from the taste buds. They are able to pick up the dissolved chemicals from the saliva and be able to transmit an impulse or create an impulse that is going to go now to, that's the function, okay? Or that's the purpose of what happens. And then it's also able to suppress bacteria. Remember, I always say that's the first level of defense. You know, when you look at the GIT also participates a lot in terms of immunity. When you ingest something, the GIT is responsible for making sure that whatever you consume, if it's infected, it will try by all means to neutralize it. If it gets into your bloodstream or it gets into where it can cause an infection, what that means it did its part. All right. You're going to find as you go down, we'll talk about even the importance of gastric acid or hydrochloric acid. That's a function. So I usually say about 90% of your immunity depends on the GIT, what you take in. COVID, cholera, all those, most of the things, they are related to the GIT, right? So take care of your GIT tract. Then also, it's able to begin the digestion of bone starch by producing an enzyme known as salivary amylase or TALI. This is able to break down that into. Also, there's another cow produce going to be produced known as lingual lipase by the Benner's gland. Okay, so this lingual lipase is where digestion begins from. All right, that's where it begins from. Let's go to the next slide. So what you are seeing here is a diagram depicting the different types of chemicals that are produced in the stomach. All right, and this is very, you need to pay attention to this part. So for example, this is a human being. This can be Charles, can be anyone, right? So now we have already talked about what happens in the stomach, in the mouth. And whatever I've been mentioned and whatever I've left out, the bicarbonate, what, and all those things that are part of saliva, please go and do a research, study those, and be able to pick up the information. Then after that, you're going to understand that in the esophagus, the food will go there, passing the upper esophageal sphincter, the lower esophageal sphincter, until in the stomach. In the stomach, there's what we call gastric pits. This is what we have zoomed in. Opening of the gastric gland or gastric pit. So now lumen of the stomach is up there, and this is the gastric. So in this gland, there are multi or different types of chemicals that are going to be produced. So what you are seeing here is source, substance secreted, and function. Okay? So there's mucus next cell that is able to produce what we call mucus and bicarbonate. What's the function? The mucus has got a function which has to do with prevention or lining the GI tract so that to prevent being corroded or being eaten up by the strong acid, which has got a pH ranging from 2 to 4 that we're going to talk about. So it protects the epithelium or the gastric lumen that you're going to see. All right. So now, as you continue, you're going to see that you're going to have what we call bicarbonate. The bicarbonate acts as a buffer. And this buffer prevents gastric acids. You know what a buffer is? A buffer is a substance that resists change. So a buffer is a substance, in this case, that resists change to pH. So it resists the change to pH, meaning when there is an acid there, you add a buffer. That acid can be able to neutralize and reduce what we call neutralization reaction. So that's the purpose of the bicarbonate. It's for making an environment that is comfortable for the contents that go into the stomach and also the stomach lining. Then as you go on, you're going to find that you're going to talk about what we call parietal cells as you go there. You're going to find that there's what we call gastric acid. The gastric acid, which is produced by the parietal cells, is known as the hydrochloric acid, which is the major culprit that is able to activate pepsinogen to pepsin. Please follow the nomenclature. Don't say pepsin. Activating pepsinogen to pepsin. Also, it's able to kill. Remember, there are some bacteria that you are able to consume with your food. So the strong acid is able to do the job. Then there's also what we call the intrinsic factor. It's able to complex with the vitamin B12 to permit absorption, right? Then when you go to what we call the enterochromaffine like cells, these cells belong to what we call the enteroendocrine system. So these are responsible for the endocrine effect. So it produces an enzyme, a chemical known as histamine. Okay? This histamine, what's the function of histamine in the GIT? It stimulates gastric acid secretion. It promotes the production of a lot of hydrochloric acid. Then when you go to the chief cells, I don't know why they're named chief cells. Maybe it's because they're the principal cells. So these chief cells are responsible for producing two things: the pepsinogen that we talked about, and also the gastric lipase. So the pepsinogen and the gastric lipase have got two functions. Okay? So number one, the pepsinogen is responsible for degrading proteins to what we call peptones or polypeptides. But their major function is to break down collagen or make the meat a bit softer or a bit easier, lack of a better word, to be acted on by the enzymes that you're going to find in the duodenum. So pepsinogen cannot act on proteins unless it's activated too. So people have got issues with pepsinogen production. Collagen digestion is affected. Gastric lipase digests what we call. So remember, in the mouth and the stomach, these two constitute about 10% of lipid digestion. Let's go to the other type of cells, which are part of the enteroendocrine cells. For example, the D cells and the G cells. The D cells and the G cells, they are known as antagonistic cells. What do you mean by antagonistic? They are opposite of each other. For example, somatostatin inhibits acid secretion, while the gastrin stimulates acid secretion. And that's what we mean. And they belong to what we call enteroendocrine cells. Let's go to the next slide. So function of hydrochloric acid, as you know, activating pepsinogen, which I've talked about, provide optimum pH for action of pepsin. Remember, pepsin works well in an acidic environment, which is a pH from 2 to 4. Then food, protein, naturation, and easy decomposition. Remember, I talked about collagen. It helps to make the meat soft, tender, and whatever, so that it can also be further broken down together with the gastric peristalsis. Okay, reducing and making the meat very tender, or the proteins or the collagen, so that they can be acted on further by the proteolytic enzymes produced by the pancreas. Kill bacteria in food in the stomach, which you talked about, the defense mechanism. Then promoting pancreatic, small intestinal, and bowel secretion. Because pancreatic promotes pancreatic small, and so it helps to prepare the secretion of these things that are needed for continuous breaking down of proteins once gastric emptying takes place. Then number 6, helping iron and calcium absorption. Remember, iron is very important for production of red blood cells, right? While calcium is important for formation of bones and also for muscle contraction. So these are the importance of what we call hydrochloric acid. So function of pepsin, function of pepsinogen, okay? Yeah, so this is a function of pepsin. So now pepsinogen is going to be cleaved with the help of the acid known as hydrochloric acid in the pH ranging from 2 to 3.5 to produce what we call pepsin. Then this pepsin will further go and act with the protein, to be more precise, the collagen, produce what we call peptone. This peptone can go back and increase also the pH. So now, function of mucus and intrinsic factor. So the mucus secretion, so soluble and insoluble mucus are secreted by the cells of the stomach, okay? The mucus cells. So the soluble mucus mixes with the contents of the stomach and helps the lubrication the lubrication of what we call the chyme. The chyme, those are the stomach contents, like for example, the food, but the volatility of the stomach is now called chyme. Okay. So now the insoluble mucus from a protective barrier against the high acidity of the stomach content, meaning it forms a protective barrier, the insoluble mucus, so that the acid does not injure the mucosa walls of the GIT. Also, intrinsic factor absorption, intrinsic factor help absorption of vitamin B, which also mentioned. So pancreatic secretion, as you know, there are two functions. There's the endocrine part and the exocrine part. The endocrine part has to do with insulin and glucagon production, which has to do with controlling blood sugar levels. While when you go to the exocrine part, which has to do with the production of different types of chemicals or enzymes. So secretion of pancreatic juice, it has two components, aqueous and enzymatic components. So the aqueous components are, for example, bicarbonate. Bicarbonate helps to reduce or decrease the pH of the contents of the chyme that is coming from the stomach once it's emptied into the duodenum. So it's able to neutralize the contents. And that's what happens there in the duodenum, so that it can be easily acted on by the pancreatic enzyme. The enzymatic component is essential for proper digestion and absorption of carbohydrate, fats, and so digestion continues. So remember, the pancreas produces about about different classes of enzymes that are able to degrade proteins, lipids, and proteins, lipids, and carbohydrates. So examples of these are trypsin, which is known as trypsinogen, there's chymotrypsinogen, there's pancreatic lipase, pancreatic amylase, etc. So in the next slide, you're going to see that you're going to go further to talk about these different types of proteolytic enzymes that are going to be degraded. For example, we have got what we call chymotrypsinogen, trypsinogen, proelastase, and carboxylpeptidase, procarboxylpeptidase. These four culprits are able to break down proteins further, but they are produced in an inactive form to avoid eating up the pancreas or tissues around that area, right? So now, once they reach the small intestine, starch can be broken down by what we call pancreatic amylase into maltose. Then triglycerides, or natural fats, can be broken down by what we call pancreatic lipase, together with the help of phospholipase and bile, to form what we call glycerol, monoglycerides, and fatty acids. And this is the range of the pH. While trypsinogen needs another type of enzyme that is found in the enterocytes or in the duodenum, which is known as enterokinase. You have heard of this enterokinase. So this enterokinase is able to cleave a trypsinogen into trypsin. So this trypsin can go further and break down proteins to form polypeptide, or the enterokinase, the trypsin can go further and break down chymotrypsinogen into chymotrypsin, which can better break down proteins into polypeptide. Then there's what we call carboxypeptidase. This carboxypeptidase has got a function, which is breaking that. It's also activated by trypsin. So the inactive form name is known as procarboxypeptidase, which is going to be activated into amino acids, peptides, and polypeptides. It's the only pancreatic enzyme that digests protein that produces an amino acid at this stage. So this is an example. Let's go to the next slide, which talks about the liver and the gallbladder. So function of the liver. So now, metabolic regulation, hematological regulation, and production of power. When you look at this, there's got a metabolic aspect, meaning the food particles that you eat or the food that you eat, it's going to be transported to the liver via what we call the hepatic portal vein, which is able to either catabolize or anabolize, meaning building up or breaking down. That's what it does. Okay. Then hematological aspect, remember plasma proteins production, also removal of RBCs. Those, remember, RBCs have got about 120 days lifespan. So after that period, damaged red blood cells can go to the liver and be replaced, and iron can be taken from that and bile to go and form other things. So I want you to go and study about the process and mechanisms that happen in the liver. The production of bile, as you know, comes from breaking down of RBCs. So again, when you go to the secretion of small intestines, the secretions from the duodenal glands and intestinal glands, the secretory volume, which is about 1 to 3 liters a day, the book that you're using. So it contains inorganic ions such as mucoproteins, IgA, which is immunoglobulin A, and various enzymes such as enterokinase. Remember? So because small intestines include also the duodenum. So without enterokinase, we cannot commence the breaking down of function. Protective effect by mucus, digestion by enzymes such as peptidase, sucrase, lipase, and dilution. So in the small intestine, there is a lot of fluids that are produced, like water and what for dilution, which when there's oversecretion can cause diarrhea, right? Either osmotic or secretory. Then when we talk about digestion by enzymes, remember I've got what we call brush border enzymes. And these brush border enzymes, what do they do? They are the ones that complete the digestion of protein. Digestion of these disaccharides or complex products into monomers, from polymers to monomers. That's what happens there, right? So examples that I've given us is sucrase and lipase, but we'll talk about these things further under digestion and absorption. Secretion of large intestine. So in the large intestines, as you know, the colony, there's what we call colonic alkaline secretion, which visualize acidity produced by intestinal bacteria such as E. coli when they're breaking down things. The secretion of mucus, protection, lubrication of fecal matter, because as the feces are moving in the large intestines, there can be injury. So there's some mucus that is produced, which causes lubrication. All right. So you find that you have good dysentery, you find that the mucus will be coming out. That's what they're basically talking about if you have gone to the toilet for a long time. Then vitamin B and K absorption made from bacterial flora in. So this is the end of the discussion. Next time, we'll look at digestion and absorption. Thank you for the time. God bless.